me know whether it is visible or not.
Yes, sir, it is visible.
Right, thank you. So, in the group, I have seen there are a lot of participants, right?
Yes, sir.
300 plus participants are there.
Yes, sir.
So, can I start the lecture or should I wait for?
Sir, we can start the lecture.
Okay. Thank you. First of all, so thank you to Bahana College, who has organized this very important training program on bioinformatics. So basically, Institutional Biotech Hub, Bahana College, they were organizers of this uh, training program. So it would have been a rather good experience if it is a physical workshop. But anyway, so COVID has given us a lot of tools, a lot of techniques through which we are now comfortable in up to some extent online also. So I'll start the lecture. Okay.
Okay, sir.
Yeah, I'll request all to mute their microphones, and if they want to interrupt me with any questions in between, they are most welcome. So we'll be going from the very uh, basics of computer-aided drug discovery. So it's a very vast area, right? So very vast area. It has lot many techniques, tools, and resources are involved. High computational power is required. Okay, so how the entire that scenario is being changed by bioinformatics, that I want to just give a glimpse to you. So 1.5 hours is basically, it's a CADD is a topic for an entire workshop actually. So whatever we will cover, and I hope that this 1.5 hours should be interactive one. It is not like that I am only saying and you are listening. Somebody may interrupt if any question arises in between. Okay. So good morning all. In your screen you can see there is a cup of tea, right?
Yes, sir.
So which we take, most of us we prefer to take in the morning, right? So have you thought tea sometime as a medicinal drink or medicinal plant? Maybe most of you have already familiar that tea is basically is a health drink, right? So it contains a lot of polyphenols, those which you term as catechins, epigallocatechin gallate, EGCG. Then many other catechins are there in the black tea as well as in the green tea. Okay. Then after that, so after this lecture is over, it will be the lunch time, right? 12:30 or something like that. So we take a lot of things in our daily life to survive. Okay. So knowingly and unknowingly, along with the proteins, along with the carbohydrates, along with the vitamins, we take a lot of other medicinal compounds. We consume a lot of other medicinal compounds, which basically keeps us healthy and safe from many diseases. Okay. Now, the today's lecture I will be focusing on basically the hidden treasure which is there in our medicinal plants, what we consume in different forms, like in form of tea, or in the form of, say, wholesome lunch, or say in the form of different Indian spices. Okay, so spice, whatever you say, it is black pepper, piperine comes to our mind, right? Then gingerol from the ginger, curcumin from the turmeric. Okay, like that, these medicinal compounds are abundant in these food materials what we consume in our day-to-day life. Now, the amount in which we require, suppose we know that the green tea is good for so many diseases, like you can say it is good for the arthritis, good for, it has got good anti-aging properties. But have you thought that how much green tea we have to consume in order to get those adequate amount of compounds? Like suppose EGCG is one of the antioxidant, very strong antioxidant available in green tea, right? So in order to get that green tea, EGCG in the required amount, we need to have many cups of green tea during the 24 hours cycle, and which is not possible, right? Because there are so many other chemicals available, phytochemicals available, like caffeine is there, which can cause gastritis. There are other compounds in the medicinal plants which may be having some adverse effect in our metabolic activities. So it is not like that something we are getting from the nature means if we take it as a food, okay. But if you want to take it as a medicine, the required dosage form, required amount is very essential to be remembered. So that is why what people are doing, that we are searching the required chemical, required phytochemical in a particular medicinal plant. Suppose we know that tulsi is good for, say, cold and cough. But what are the exact compounds which is working for giving us the benefits? From where they will come? From the phytochemicals. So day by day, the diseases are increasing, right? Newer and newer diseases are coming. So we need drugs. We need new molecules into the clinic. So from where we can get those new molecules? Definitely, nature is the first choice, right? Natural compounds. From where we'll get abundance is there in the plants. In the natural context, in the plants, in many bacterial species, many fungi species, right? Many medicinal compounds are available. So how to select those medicinal compounds which are essential for a particular drug, so for treating a particular disease? Say, for example, we know that some of the medicinal plants are very useful. Traditionally, it is being used, say particular disease, like as I have mentioned about the tulsi, or as you can say that the Aratoda vesica, or turmeric, or whatever. So people are using traditionally, so from our grandparents, great grandparents, from our forefathers. We are getting the legacy that we should take this. If we suffer from, say, indigestion, we should take, say, ginger, right? But now it is time to identify that what are the compounds that is creating the difference, which are creating the magic for us. Okay, so that what will be the benefit of that? Number one, we can get the right compound in the right amount. Number two, we can ignore the unnecessary compounds, unnecessary pigments, or whatever available along with the naturally occurring the active compound or the required would be drug molecule. Means we can nullify, we can remove some of the unnecessary phytochemicals, and we can focus more on the actual compound which is required to be developed as a drug. Okay. So why we take the plant-based products? So I'm speaking as a layman. We take plants as a food, right? We take plants as medicines, traditional medicines. We take plants as nutraceuticals. We take plants as functional foods. Okay? Functional food means those plant products are being used in some protein supplements, right? We can use the plants as intoxicating agents. Many plant products are being used as a intoxicating agent that tobacco products are plant products. So basically, there are so many big list of materials what we consume that comes from the plants. Okay. And neem is used for what purpose? Okay. But to be more precise, so traditional medicinal plants are being used for many ailments. Why it is necessary? Yes, please. Siva Chetri. Any question? Siva Chetri, you have raised the hands. Yes, please. Any question? Am I audible?
If you have any questions, please unmute yourself and you can ask. Okay.
Yes.
Maybe by mistake he has raised his hand.
Okay. No issue. So the beeping sound comes actually in between. Yeah. No issue. So traditionally, medicinal plants are being used for treatment of many ailments, right? So all of you, we know. So why it is necessary to know about the specific active compounds, secondary metabolites, etc., present in the plant? So we may not get the adequate amount of active compound, as I have mentioned, that you may have to take 15-20 cups of green teas per day to get adequate amount of EGCG in your diet, right? And secondly, washing away of the unwanted compounds. So we can wipe off the unwanted compounds which are basically not necessary for our body. So that is why how we can do that, how we can get that exactly what are the molecules present, what are the active molecules present in that plant, and where it will be useful. So basically the simple answer is the biochemical studies, right? So how we can do that? So you know that we can extract the compounds in different solvent systems, right? So after extraction of the extracting the compounds, we can take the help from the separation techniques like high performance liquid chromatography, thin layer chromatography, HPTLC, okay, high performance thin layer chromatography. So GCMS, gas chromatography and mass spectroscopy together we can use. Then paper chromatography. So there are so many separation techniques are available that you already familiar with. So generally what we do is GCMS and the LCMS. The most commonly it is being done. So if you want to do the analysis of a particular compound, particular plant, the compounds in a particular plant, okay. So we'll have to do the proper extraction. Then we have to send the samples for GCMS as well as LCMS analysis. Right? So anybody want to start any work on medicinal plant, they can follow this protocol. So you don't have to have GCMS, LCMS at your lab. There are hundreds of organizations, institutions, labs which outsource their services. You can outsource your services to those companies, right? So GCMS and LCMS, basically GCMS, it detects the volatile compounds, and LCMS for the non-volatile compounds. So in the screen you can see the different kind of peaks you will get, and from the peaks, these peaks, GCMS spectra can be analyzed, and the compounds one after another. This is LCMS spectra. So liquid chromatography and TOF mass analyzer, time of flight. So ultimately you will get some of the peaks, and the peaks are basically, these are identified to be some of the active compounds through those internal databases of the software, or there are many ways to analyze the GCMS and LCMS spectra and get the compounds identified. Say your GCMS and LCMS result, after analysis you will get the names of the compounds like this. Okay? So you may not get the structure, okay? But you can get some idea that what is basically this particular compound is. So I'll show you that from here, say this is dimethyl fumarate. So we can simply go to one database. This is NCBI database. So where there is a database called PubChem compound. So in a PubChem compound database, we can take the name identified by your GCMS and LCMS analysis. Suppose dimethyl fumarate. Just you can search dimethyl fumarate. And you can see the structure and the properties of the dimethyl fumarate here. The PubChem compound database. Kindly note this. So I'll show you. So there is similar analogous database maintained by EMBL, European Molecular Biology Laboratory, which is known as CHEBI. So same compound, say dimethyl fumarate, can be searched in this database also. See, this is the compound. Okay. Fine. So we can use these compounds and we can build up a library of the compounds which are available in this particular plant. Okay, so we have got lot of other properties, like one important is canonical smiles and isomeric smiles. So these canonical smiles and isomeric smiles, this can be used for generating the structures without drawing. So there are software available through which we can regenerate the structure and calculate the properties. This is one software, BioVia Draw, okay, prepared by Dassault System. We can generate the structure, regenerate the structure from the smile string what we have copied. See? Fine. So we'll do it practically when you do the practical sessions. Now, interesting thing is you can calculate the properties on the go. See, you can see the properties has been calculated within one click. Molecular formula weight, molecular mass, molecular exact mass, so on and so forth, which are also available in this PubChem compound kind of databases. So you can see the molecular weight here and molecular weight calculated here, exactly the same. Okay. Now the thing is, see, after getting the compounds, these are some of the compounds how we can identify from the GCMS and LCMS result. So I'll repeat again. Say for example, we have a medicinal plant, right? So if we want to do any structure-based drug discovery kind of work, or we want to identify the drug targets of the active compounds of these medicinal plants, first of all, we need to do the identification of the compounds through these separation techniques and detection techniques like GCMS, LCMS, HPLC, HPTLC, like that. So after getting the compounds, when GCMS and LCMS will give a result, which are basically IUPAC name, or in some cases, some common names will also be given. So those names can be searched in the phytochemical databases or chemical databases. Sorry. I'm not very well, so please bear with. So we can search the same thing, the compound in PubChem compound database or a similar database that is called CHEBI database. So these two databases are one of the very beginning point for doing any kind of structure-based drug discovery research. Okay, so Northeast, basically Assam, is full of medicinal plants. There are hundreds of varieties of medicinal plants which are still to be explored. Okay, so this way we can identify and we can make a list of the compounds as well as the structures like this property, and we can prepare our own data set ready for doing the analysis. Now we'll take examples of two different diseases. So how these compounds can be screened against some diseases. Say one plant that is traditionally known that it is used for treatment of the Alzheimer's disease, which is a non-infectious kind of disease. Another one we'll take, which is basically active against infectious diseases, bacterial infection, viral infection. In this case, I'm taking the example of COVID-19. So COVID-19 is back with JN.1 strain identified in India again, but there are mutations going on, and we'll talk about that also. So how cleverly the viruses, they pass through this kind of pathways which are targeted basically by the drug discovery researchers. So we'll take two examples. One is non-infectious disease, another one is infectious disease. Fine? So suppose you want to start your research against Alzheimer's disease, which is a very common old age disorder in today's world. So we have to see the Alzheimer's disease pathogenesis first of all. Okay, the entire disease pathogenesis we have to study before going to target any protein, any enzyme as our drug targets. So beta amyloid production pathway we have to see the presence of neuropathological structure, neurofibrillary targets. So this we will discuss little bit, not all. Okay. So, and there are many epigenetic targets also available for diseases like Alzheimer's disease. Now, after knowing the pathogenesis of the disease, okay, second part will be to identify a very important pathway of that particular organism. Say in case of Alzheimer's disease, this is ultimately the human only, we are doing for human. So we have to identify the pathway, metabolic pathway, signaling pathways, reaction participants. What are the enzymes and the proteins which are participating in the disease pathogenesis, relations between genes, enzymes, and reactions. So in this case, to identify the drug targets, that is a very massive and tedious work. And we will be in the safe side if we follow the peer researchers' works, where there are studies like gene knockout studies, okay? there are nowadays the artificial intelligence being employed for identification of the disease targets. That itself is a massive, massive chapter which basically identifies the actual pathway which is involved in that particular disease, and in that pathway we will be targeting some enzymes, some proteins as the targets of our probable drug candidates. And probable drug candidates are those candidates which we have identified in the medicinal drugs. Correct? So there are so many pathway databases are available. After getting the literature reviewed, after getting the literature searched, we have to use our intuition and brain also, knowledge also, to search the right pathway, right enzyme. That if we, just I will show you, say, example, regulation of beta amyloid production pathway and its epigenetic alterations. So in these pathways, in these kind of pathways, there are certain important enzymes, okay, which can be blocked. If we block that enzyme, suppose that beta amyloid production pathway is disturbed. You understand the point? Say, we have to identify such a target or such a protein or such an enzyme which is very, very important for this particular pathway. If you are targeting this beta amyloid production pathway, which is associated with the Alzheimer's disease. So we can target that particular protein. We can see that involvement of that particular protein or the enzymes, and we can go ahead with the rest of the procedures. Okay, so we'll come to these epigenetic modifications little later associated with Alzheimer's disease. There are many epigenetic regulators available which are also being identified as successful drug targets. This is the Alzheimer's drug development pipeline. So different enzymes, amyloid, beta amyloid pathway has been targeted. This red that rectangular shapes which were available in phase 3, some are there in phase 3, some are there in phase 2, some are there in phase 1 clinical trials. Okay? So there are symptom reducing small molecules, disease modifying biologicals, disease modifying small molecules. There are 3 categories of medicines are being trialed against the Alzheimer's disease. Some are there in phase 1 trial, some are there in phase 2 trial, some are there in phase 3 trial. What are these phase 1, phase 2, phase 3? We will discuss. Okay. Basically, these are different phases of the clinical trials where we fix the required dosage form, the mode of administration of the drug, etc., etc. So we'll come to this picture once again. Now, from all these studies, peer researchers have identified that from the terminology of the common Alzheimer's and related ontology researches, there are some of the protein potential targets for Alzheimer's disease at the early stage and late stage clinical drug development. Okay, so amyloid is one, tau protein is another, apolipoprotein E, ApoE, Okay. Sorry, lipoprotein receptors, neurotransmitter receptors. So there are hundreds of potential targets which are already being identified by the peer researchers. We don't have to work on this. Just we have to do the strategic literature review. Then after getting the name of the target, say you have fixed that we will use this pathway, we will target this and this kind of enzymes. Then next phase is getting the structure of that particular enzyme or the proteins. So this database, protein data bank, RCSB protein data bank, can help you in getting the structures of the proteins. Okay. So how we can get the structure of these proteins, that we will see. Now the thing is, from where we will select the probable drug candidates. So probable drug candidates we will select from the plants which are used in traditional medicines against Alzheimer's diseases. Say, in those traditional medicinal books and literature, we can get many clues, different clues, that this extract can be useful, this plant, that leaf boiled in water is useful, and so on and so forth. There are so many clues we can get from the traditional medicines. So here I have put a list of 16 different known medicinal plants which are almost known to you: Curcuma longa, okay, Piper nigrum, okay, Brassica nigra. So these plants, suppose we have identified from the traditional knowledge base. Now what to do next? Will you do GCMS, LCMS for all these plants to get the compounds? So basically, which is not possible. If somebody has already identified, then why we will go for GCMS, LCMS again? So there are databases for that. So definitely we'll do GCMS, LCMS for confirmation, or say if no clue is available, then we cannot go for, no clue is available, then we can go for GCMS, LCMS solutions. But before that, we have numbers of databases available. Now, one is very important database is Dr. Duke's phytochemical and ethnobotanical database. Okay. So this is maintained by US Department of Agriculture, in the official website of the United States government. Say we need the all entries for a particular plant, say any scientific name we can search here. So Tulsi, holy basil. There are 29 chemicals available. Okay, there are 29 chemicals available in this particular plant. So, out of all these 29, there may not be unique compound for Tulsi. So, suppose ascorbic acid, it's a very common compound, right? There are some fats, glycosides, so which are not essential for us, which are not important for us. So out of it, suppose oleic acid. So from this kind of database also, again, you are getting the names of the compounds. Then again, you have to take the help from these databases like PubChem compound. So this is the structure of that oleic acid. Fine. So like that, we have to build up our ligand database. If the plant is not at all available in these kind of databases, then we have to do the GCMS LCMS analysis of our own. Then rest I have already mentioned. And there are databases like PubChem compound, there are databases like CHEBI, from where we can get the specific information about the chemical compounds which we got through the searching in Dr. Duke's database or in the other databases, and if not there, through GCMS LCMS analysis. So ultimately, whatever it is, we have to collect the structures, proper properties of the small molecules. So for doing the computer-aided drug discovery, so till now, what we have got is we need some small molecules, maybe plant origin, may not be plant origin, okay? That is different thing, but I am focusing on the plant origin small molecules. So we have to combine the information of say 30, 40 plants, whatever is found to be active, say list of the compounds available in these 16 medicinal plants. Suppose we have collected and we have listed them properly with proper property calculation and other things. Then comes, so we'll go to another disease, but before that, before doing the actual filtration of the molecule, so we'll not take each and every molecule what we get in databases or what we get in GCMS analysis. We won't require that. Okay, it is not required. So we'll get only some selected compounds, and before that we can do a mass screening of the compounds for drug likeliness properties. So whether The plant compounds may also be toxic, right? They may have some solubility issues, okay? They may have some other issues which are not favorable for becoming a successful drug. So those compounds we have to wash away at this stage only. So unnecessarily, why to take it forward for docking and other thing? Fine. Then we go for another disease. So we'll come to those processes again and again. Okay. Because our aim and objective is to see the tools and techniques which are prerequisites for computer-aided drug discovery. That is the theme of the lecture today. So, but I'm giving two examples. That is one Alzheimer's disease, that is a non-infectious disease. This is COVID-19, infectious disease. So you all know that a pneumonia of unknown cause detected in Wuhan, China. So first reported when, so, okay. But COVID-19 is caused by SARS-CoV-2, a RNA virus, which is a new or novel coronavirus. Okay. So basically, the controlling the viruses through vaccines, through medicines, it is not very useful. Okay. It is not very useful because whenever the molecule we deploy as a drug, okay, the viruses are so clever that it may change their target protein structure through accumulation of mutation. Viruses are very fast mutating agent, you all know, right? They can change their code protein, they can change their target proteins, everything they can change within very, very small time. So it is sometimes becomes very difficult, okay, to trap the viruses with some vaccines, with some small molecules. But there are strategies which are being followed worldwide. So where multiple drug targets are targeted by single compound. So in that case, if one protein get mutated, the small molecule can bind to the other one. If second one fails, third one, like that. So in the absence of new targeted drugs and vaccines against SARS-CoV-2 at present, the choices for the effective treatment are limited. It was limited and it is limited. So by applying the principles of computer-aided drug discovery, we can contribute by the following ways. The screening of already available drugs used for treatment of other diseases, which was done. Many of those antibiotics were screened, so you know that the rosiglitazone, then many failed also, like remdesivir, okay? Those are called repurposing of the drugs. Then another way is discover and identify new medicines from different sources like microbes, plants, etc., etc. So this is the right side panel is our focus panel for this kind of exercises. So discover, identify new medicines from different sources like plants, microbes, etc., etc. And most importantly, why CADD is so popular? Because you don't have to come to the contact of the virus physically. Okay. So the virus name is SARS-CoV-2. Those red marked names are basically well recognized disease targets. Main protease, 3CL Pro, RDRP. Okay, there are so many PL Pro. There are so many drug targets have already been identified. People are working on that, and these basically helps in the viral propagation. So that means our aim is to limit the viral propagation through administration of the small natural compounds. So natural products through novel drug development exercises are basically developed against these kind of drug targets. Then the natural molecule binds to that, say, main protease. It can block their activity, and it can seize a particular pathway or a particular metabolic pathway. Okay, so which will lead to the controlling of the viral propagation. And as I have mentioned, that viruses are very, very clever agents. They can sense the target proteins. Say you have designed a drug screened a drug against PL Pro or 3CL Pro. By the time you administer the drug, it senses its drug protein. The active site where it was intended to bind has been changed. How? By mutations, accumulation of the mutations. But still, if we take 2-3 protein targets at a time, against which we administer a small molecule which can bind to all the three drug targets, then the success ratio of this drug discovery programs becomes manifold. Now come to the main part. So what are the steps in computer-aided drug discovery or CADD? So as I have told, target identification. What is the target? Again, I am repeating, the target is the protein or the enzyme or some DNA molecule which are available either in the pathogen or in some of the targeted host organism. Then we do the structure-based drug designing and ligand-based drug designing. There are two different ways. In a ligand-based drug designing, during the QSAR and ligand structure information ligand has been designed against the active site, and QSAR, quantitative structure activity relationship, and QSPR, these techniques are applied for designing a proper molecule which can fit to the active site of a protein target. Now what is happening in this process? AI has been applied, artificial intelligence is applied. So you give the properties that I need some compounds with these properties, this site group, that site chain, etc., etc. The AI, the generative AI, actually can increase the probability of designing a better drug candidate increases. The probability increases with the application of the new techniques like generative AI. Okay. So that is ligand based. So we are not talking today about the ligand based drug discovery program, but we are talking about the computer aided drug discovery program where we need the target structure. Again, I'm repeating, what is the target? Target is the simply at this moment we consider that it is an enzyme, a protein, right, which is important for the disease pathogenesis. Then we do the ligand docking. Ligand docking. So from where the ligands will come. Maybe from the databases like PubChem compound and other things. Maybe from the plant sources. Maybe from the bacterial sources. Okay. We have to build up a big library of probable compounds which can bind against a particular target or 2-3 targets which are selected against a particular disease. Then what we will do, in the process of ligand docking, what we do basically, just I'll show you. Again, I'll come back. We try to fit, this is the active site. This is the active site of the protein target. This is the ligand. Okay. You see how beautifully it is binded to the protein target, more specifically to the active site. See how the structure will bind, how the compound will bind. See, the conformation, right molecule in the right site. So if we want to try to do the fitting computationally, that is basically called the docking. Means you have thousands of keys and you have one lock. Now you have to search for the molecule, that means the key, which can unlock the receptor protein or the active site. Do you understand? Now there will be many keys which will open the lock, but not in a very efficient manner. You have to do very hit and try to do the opening of the lock. Okay. But there will be a few keys, 1, 2, 3 keys, which will very swiftly open the lock. You select those things. And that is called selection of the ligands. From the ligands, those process through which we have selected the 3-4 compounds which can be termed as the selected ligands or the lead molecules. That process is known as the computational docking. Okay. So after docking, say one particular compound is binding to the other particular compound, that the protein target in the active site, one small molecule is binding. Whether the binding is properly being done, whether the binding will be remaining intact in that portion, that is basically signified, proved by a process called molecular dynamics. So we can do molecular dynamic simulations using very famous software like Desmond of Schrodinger, Chromex, right? There are ways through which we can predict, okay, this is bind to this target, but how good is the binding? Okay, those things we have to see. Then we have to do the lead optimization. And suppose 10 compounds we have selected or 5 compounds we have selected from the thousands of keys. Now all may not have the right nature or right properties to become a successful drug. We have to do the ADME tox calculations again. Absorption, distribution, metabolism, excretion, and toxicity, ADMET. Then we can do the lead optimization. We can change the solubility pattern and other things. Nowadays, in the lead optimization process, the generative AI is again being applied, and we can finally do the identify that this ABC, these are the 3 probable molecules. We can say probable only through as we are doing computationally. We can just term it as probable. This is the probability. These can bind to this target protein of SARS-CoV-2 and may lead to controlling the growth or propagation of the virus. So that is in a nutshell, that is the story. So if you have any question, please unmute yourself and raise the question. Am I audible?
Yes, sir.
Yeah.
You're audible. Anyone have any queries?
Okay. So again, I'm repeating the steps in computer-aided drug discovery. So one side, that is selection of the drug targets. From how we select the drug targets, mostly depending on the previous literature. So at this moment, at our organizations, some of you may have the facility for drug target identification studies, but if we think for a total computational work, then we have to depend on the previous literature. Then get the structures from the drug targets from RCSB protein data bank. Then prepare the protein structure for docking using the software like CSF Chimera or other thing. That means this is part, left part is the for protein and right part is the for small molecules. So selection of the small molecules for screening. These are the small molecules, say, ready for screening against a SARS-CoV-2 viral protein. Fine. Now, what you need, sorry, what you need at this moment, you need the 3-dimensional structure of the protein as well as 3-dimensional structure of each and every ligand. So who are the ligands? Where are the ligands? These are the ligands. And this is the protein, target. This is the target protein. So one small molecule, one protein, matching. Matching hoga ki nahi hoga, whether it will fit or not in the active site. If it is fitting, how, in what degree it is fitting, what is the docking score, what is the energy, total energy of the protein ligand complexes. These are being calculated in the process called docking. So when there are thousands, millions, 10 millions, 20 millions compounds are available for screening against a particular drug target, then the normal computation is not going to help you. So in that case, nowadays we are using the HPC's high performance computing system with some software based quantum simulator. So you know that one another generation is computer is evolving very fast. There is quantum computing which will take over all the computation calculations in near future. So quantum computing, that hardware we may not use, but there are some software which can increase the power of your desktop, power of your laptop, power of your GPU enabled HPCs. So these are basically when we do in a massive high scale calculations, massive data. This is already a big data analytics, right? So this massive massive compound library, the small molecules, these are one after another. This is being screened against a protein target. This is basically also referred to as, if the data is very huge, there is high throughput screening. Okay, so high throughput screening, and then extra precision docking also we can do for precise selection of the small molecule which will be better fitting to our proteins. But the prerequisite is, I am telling repeatedly, the prerequisite is you identify the protein target properly, you identify the small molecule compounds from where you will take the database compounds, you will take some plants, okay? Then the protein as well as the compound, the small molecule, protein and a small molecule both need to be there in their 3D structures. 3-dimensional structure will be required for both protein and the ligands. So in case of the SARS-CoV-2, again I am coming back to that. The following are the potential drug targets being used commonly by the researchers throughout the world. One is main protease, there is 3CL-Pro, RDRP, 2'O ribose methyl transferase, so on and so forth. How the drug targets are determined, that I have already mentioned, that it is through literature survey we have to do, or otherwise we have to work in the wet lab to identify the drug targets by mutating the genes or gene knockout studies. And there are so many molecular biological experiments through which we can identify the targets for a particular disease. So what information is required to be available to select a protein drug target for a study? Sequence of the protein will be required, if not the structure. Structure of the protein from protein data bank. If it is not available in the protein structure database, then we can take the help of that Google's DeepMind AlphaFold. Okay, it's a AI generated protein structure database, which can be very confidently used for getting the structures of those proteins for which the structures are not available in protein data bank. Most of the structures you will get in protein data bank, but for many of the structures you may not get the structure. So in that case, we have to do the homology modeling. But nowadays, we don't have to go for homology modeling. Why? Because the Google's DeepMind, DeepMind, Okay, Google's DeepMind has developed one AI-based technique, AI-based homology modeling system, which is known as AlphaFold, which has solved almost entirely all the protein structures. That means homology modeling is almost not required nowadays. So if you do not get in protein data bank, then we can go for searching the databases like Google AlphaFold databases. Okay. So this is the protein data bank. So how to download the protein data bank structures? Just I'll show you. I'll give one assignment to do during the practical classes, which will be guided by the respective faculty members from organizing institute. So, but quickly, I want to show how to get the structures from protein data bank. Any protein. The URL is rcsb.org. So any protein name, suppose human hemoglobin you want to download, 1A00 is the okay. So before downloading the files, you have got lot of file options to download. So we have to download a simple software called Resmol. We'll start with Resmol. Or Reswin. Okay. So this is a three-dimensional structure visualizer. So the structure files which are available in the PDB format, I have downloaded it. This is open. You can see. So there are 4 chains in this hemoglobin molecule. So this is one. You can visualize, you can see how many alpha helices are there and this, that. Then another one is Chimera. Okay. It's a little bit high-end software. So this is the structure. Here it is little bit more beautiful. You can color the chains. Okay. So you can see the hydrogen bonds. Can you see that's red marks? Small red bonds. These are hydrogen bonds. You can See where the hydrogen bonds are there and how the hydrogen bonds are keeping the structures intact. So these are some of the examples of some of the visualizers. This is another one visualizer. You can open the structure. This is another one. You can see the hem groups inside. Okay, so you can see the interactions. Okay, there are 4 ligands. So anyway, you can see the interactions also. So that way we can download the structures from the protein data bank and all, and keep the structures ready. So before doing the docking, we have to process the structures. Process means, say this one only we will download 6WNP. This is called PDB ID, right? 6WNP. So before doing the docking, any bounded ligand to be deleted. Any solvent to be deleted. There are tools for preparing the structures, like for docking preparation, that is known as dock prep module. Delete solvent, add charge, other things. So this is an important step before going for docking. We have to do this. And secondly, a small molecule from the plants. So what plants to be used, where we will get the molecules, that will already I have mentioned that we have to take help from the traditional medicine or any traditional use, Ayurvedic use, whatever. These are some of the examples of the plants which are being used against the SARS-CoV-2 viral proteins. So again, you have to select the structures from the PubChem compound. We have to make the 3-dimensional structure of the ligands also. As already I have shown, the 3-dimensional structure of the protein we have downloaded from the protein data bank. Then on the other hand, we need the 3-dimensional structure of each and every ligand which we want to dock with the protein. Protein is ready, as I have shown to you. Now, the small molecule you have to prepare. Small molecule, as I have told, that so we can use the online tools, like one is Corina is there. This software can be used. Or simply, this camera can be used for building the molecule, 3-dimensional structure of the molecule. So these are examples of the small molecule 3D structures. Okay, from the plants, from PubChem compound, then we design through those Corina and other software, then Chimera. Then finally we get 3D structures like this. Okay. So this 3D structure and the other 3D structure of the protein is required to get the calculations done, whether it will bind or not, this molecule will go into the active site or not. Okay. So ultimately, after docking, you get a result like this. Say this, sorry, this is the ligand. Can you see? After docking, this one, right? This is a small molecule, this is the protein. Then we can see the interaction diagram also. See which amino acid? These are hydrogen bonds which is being created by the ligand, and it is binding to the nearby amino acids. So there is a rule called Lipinski rule of 5, which we'll have to obey during the selection of the binded ligands. So if we do it for say hundreds of thousands of molecules, there will be a score. Means to get the receptor ligand after receptor ligand docking, there will be the docking score which will tell you that which compound, which small molecule is binding more favorably to the protein. So select the best binding ligands using Lipinski rule of 5. There is a drug discovery rule. Not only Lipinski rule of 5, there are Ghosh rule and other rules are available, which can be applied for selecting out the actual drug-like compound. Then perform admitox and drug-like studies of the short-listed molecules. Then we can propose that this molecule can bind to this target up to this level. Okay, that is not confirmatory. We cannot say that we have identified a drug, but we have identified a lead molecule which can be developed into a full-fledged drug in due course of time with due proper wet lab validation. Okay. So this is the summary of the experimental strategy: selection of the crystal structure of the target proteins from RCSB protein data bank, processing of the crystal structure of the target protein. How you process? We process it in Chimera or some other software. Then processing of the plant-based small molecules. Then molecular docking. Then molecular dynamic simulation. Why we do the docking? To select out the best possible binded ligand with the receptor protein. Why we do the molecular dynamic simulation? After a small molecule binds to a protein molecule, whether the binding will be stable or not. How many times? Okay. how many till how many nanosecond it keeps on binding and etc. We have to do that pharmacokinetic analysis. We have to do those analysis that whether the compound will be drug-like or not. So QSR is another option where we can modify the molecules in more details. See how in the active site the small molecule is binding. Processing of the small molecules, then docking. After docking, already I have shown this process. This molecule has to be fixed in certain positions. This is the spike protein of the virus. This is the molecular dynamic simulation of the molecules, atoms, which are available in the molecules. To denote that what exactly the binding says or binding, whether it will remain or not. Quantum mechanical models. So these are a little bit more into the physical chemistry side. So definitely we'll see, but we, as per the requirement of the workshop, we can just confine to the preparation of the small molecule and the proteins and up to certain docking exercise. So biological data is already big data. So big data analytics tool out there for data analytics today. Platforms like Nine is available. Those who are working in this area can try using this platforms. And blockchain is also being used nowadays to keep the laser. It's a distributed laser technology. In blockchain, once a data is written, that cannot be modified or so potential use cases in R&D. So that we will see, and I'll yes. Cloud infrastructure we can use. Suppose, say at your college or at your university, you don't have the facility of high performance computing. Say you want to do one experiment. So for a specific time, say 2 days, 1 day, 5 hours, 5 minutes, 5 hours, 1 hour. So we can take some very high performing computing system hired in AWS, Amazon Web Services, or Microsoft Azure, Google Cloud. These are available. So we can pay, but we have to be very careful while using these clouds, because if we don't know and keep the machines running for longer time, so huge amount of bill will come at the end of the month. So that's all from my side in the theoretical part. I'll give one structured part by part to do work, so which can be held by the experts in the Biotech Hub, Bahana College, because I have some preoccupation, as I have already mentioned, in the second half. So it will be very nice if you can interact. So I could not deliver very properly because I'm not very well today, but whatever. I'll say that many of you can start working, although not individually. You can collaborate with each other. The colleges, the universities, they are coming up with good uh, cross-platform programs in NEP. So take the advantage of that and use the facilities given by those high-performance computing systems of the IITs and other good institutions, and you can do the work, even though you don't have the setup. Okay. So thank you so much.
Thank you so much, Dr. Shravanda sir. It was a very nice and very informative session. I hope participants have also come to know a lot about molecular docking, protein structure, and so on. So I'd like to request the participants that this session is open for discussion, as our esteemed research persons will not be available in the second session. So if you have any queries regarding whatever we have shown us in this first session, you can please unmute yourself and you can ask.
Yes. Or you can put the questions in the Google, that WhatsApp group also, if it is not coming at this moment to your mind. So that I am there and available in the WhatsApp group also. Okay, I'll be sharing the assignment and the files through that WhatsApp group only. Okay.
Okay, so we have created one Google Classroom for submission of these assignments and all, because yesterday also Dr. Pankaj Setia sir, he has given an assignment. So, dear participants, we will, those who have registered, through your registered email ID, we will send the Google Classroom code. Please join the Google Classroom and we will upload all the assignments, and you try to do it from your end. And if you might in the process, if there comes any query, you can put it straight in our WhatsApp group itself, and also through mail also, because our resource persons, they are available in that WhatsApp group as well. So that is what we wanted to share with you from our end. So now it's your time. If you have any queries, please quickly put it. You can also put it in the chat box. Thank you.
Okay, if no question is available at this moment, we can end the session, right? So,
Yes, sir. Thank you,
yeah,
sir.
I once again thank the team of Dr. Sangeeta Das, coordinator of the Biotech Hub, Bahana College, and all other associated faculty members and students, and especially to the participants from different part of the country who has joined the program. So thank you so much.
Thank you, ma'am, and thank you, sir, so much. Now I like to end the meeting with a formal vote of thanks, and I request Miss Malika Bora to deliver the vote of thanks.
Thank you, Zakaria. Ladies and gentlemen, very good afternoon to all. It's me an immense pleasure to deliver the vote of thanks for this event.
I think there is one question in that chat box. Maybe we can answer that first. Yeah. From Rahul Lodha, he has asked one question: how cameras and
Yeah,
orthodox
yes.
is used effective? Yeah.
So basically, without Chimera and Autodoc and MSR drug discovery work, means without using, if you do not want to use any commercial software, okay, this Chimera and Autodoc is the only thing which we can use very confidently. So in the Chimera, basically we have to prepare the protein structure as well as the small molecule structure. And in the Autodoc can be used in the Linux environment. If you are not familiar with Linux environment, you can use some pipelines like Pyrex, Pyryx, Pyrex, that is available for Windows also. So you can use the Pyrex and use the Autodoc and the Autodoc Vina. It's very simple. So you can try learning the Autodoc and Autodoc Vina by seeing the different YouTube videos and tutorials. It's not very difficult. And both of them are free software. So you don't have to pay anything. Just try it out once. If you have any issues, you can contact us.
Thank you, sir.
Okay, now I continue.
Mike.
My special thanks to our returning session for accepting our invitation and as well as for being with us and honor this event with this inspirational talk. And I would like to thank our beloved coordinator of the Right Heart. And last but not the least, our beloved participants for making this event successful. Once again, I thank one and all present here. Thank you.
Thank you. Thank you.
Thank you.
Thank you,
Thank you, sir.
everyone. Thank you so much.